will guide you through the experience that maybe this person already has recruiting, participating in the clinical trials or receiving a certain line of treatments, dealing with some side effects that, could be very helpful for patients. The other program is our Caregiver Connections and Patient Perspective. These are, monthly regular meetings that are peer-led. So we have caregivers, we have patients, and it's really a safe environment for patients and caregivers and families to participate and to share whatever they feel they want to share. It's a very candid discussion. It's on a regular basis, and like all of these type of supports are very well received by the community and very well and very much needed. So we have those two programs. We had an initial, I think 6 months we're in. Response is great, but just want to make sure that you have that resource available and you can pay, you can point your patients or anyone who is interested to know that these things exist. Other thing we're doing very briefly, we are developing, for instance, specific resources. For instance, we created one for financial resources, how cancer impact work, understanding, the employment laws, determine the right options for patients in their career during and after the diagnosis. I think was one of the things that, feedback that we got from patients to learn more. But also creating other type of, materials like, the symptoms, knowing the signs, and more of this is coming all the time. So if you have any feedback on something that we could use, this, we can create new things, but these are already available, and, you can download it. They're in different formats, digital, and also you can order, paper. Another thing that we've been doing also is trying to put out important discoveries out into the public, make more knowledge of great discoveries that are being made that patients should be aware. This is a partnership we did with Christy Johnson's Foundation, and we target specifically exercise and sickle cell trait from the study from Pavlos at MD Anderson, where we actually, created a very lay, piece that explains this risk factor in a very simple language that is engaging and understandable by patients, and they can refer to. So we're going to push this more because I think it's going to be important for this specific population to understand about this risk. And we're going to planning to do more of, the great things that we're learning every day about kidney cancer. So here is an overview of, let's say, all of our affiliates program. Now we have 6 currently on, and the advocacy partner organization that we're working with on specific projects, like, the Immerman Angel that I mentioned before. Another very important piece that I want you all to be aware is our virtual patient symposium, which was very successful. We ran it in September, and there were these are lectures done by professionals with a target audience, of course, of patients, explaining what are, practical things like, understanding your pathology report, or, what happened, behind the walls of a tumor board when your tumor is discussed, when your case is discussed in a tumor board. But also other, session about nutrition or supportive care were very, very well received by the feedback that we received from patients. So we're planning to do more of this. All of the material is available on demand on our website. So if there is any of these topics that you want to refer to, just know that this is available on the website. Another program that started as, very 2 years ago at IKCS, but now has really matured into a full-fledged program is the Women in RCC. I want to highlight the first meeting we had officially for the program that now is a year-long program. In July, we had a very nice attendance, and we have also follow-up at this meeting. So I hope you also reserved your spot because, you cannot walk in. You have, choose this. So for those who actually reserve their spots tomorrow morning at 6:30 in this room, we'll hear, you know, about this group with, you know, a guest speaker, Hannah Riley-Bowl from Harvard Kennedy School. And we also have other, you know, meetings and activities that are stemming out of these discussions that are like important for, you know, the career, but the development of women in RCC in our community. So here is a QR code if you want to stay updated. We have a website, but you please do, because it's not only women, but this year we also have allies, and we want, you know, to hear feedback and by the entire community. Another very important piece that we do every year is the KCA Award Program. Here you see the lineup of the awardees of this year. We were very pleased, and we awarded over $400,000 in grants. This year we had specific calls and support, in particular with specific affiliates and other organizations that supported specific topics. We want to go down the road of, you know, continuing this partnership with affiliates that are, you know, passionate about bringing or keeping a line of research in a specific subtype, but also supporting, you know, the general, you know, research that is happening in kidney cancer. When we talk about access to data, here is a slide where you probably heard about the Data Federation project that KCA started exploring. It came out of an idea from a think tank 2 years ago. And the basic idea is very simple. How can we gather access to data in a way, in a safe way that, you know, it doesn't break privacy, but is also secure, and allow us to have access to quality data directly from the hospitals. So the federated learning practice is something that Array Insight, also known as SAIL, has on their, you know, capabilities to do. And we were exploring the way of federating different hospitals. The data never fly out of the firewall of the, so this is the beauty of the technology, but the data descriptors are able, with federated learning, to answer the questions and the queries that we want to do. So I analyzed this, you know, the last 6 months I've been at KCA, and we're trying now to really break the single blocks that we need in order to kick this off. So the idea is to create 2 centers as a pilot for, you know, a proof of concept. And the 2 centers are Vanderbilt and, you know, University of Wisconsin. If we can get this off the ground, we'll probably look at 2024 for an expansion to add more hospitals. Of course, the beauty of this is that the more we have, the better, because we get more access to data. In concomitance with that other initiative, we're also exploring, you know, other opportunities to access data. And nowadays, there are so many commercial options that give you access to real-world data or real-world evidence that are, you know, claiming whatever, you know, in terms of, you know, capabilities, AI, quality, structure, anything. Here in this picture from this article, I just want to highlight how these systems normally work. Is a mix of getting, you know, data from the electronic health records or other sources, and then structure it in a structured or semi-structured way in order for systems to map them and then you reuse it. Of course, there's a lot of bias. There's a lot of, you know, different vendors. So we are now in talks with companies and trying to see what is the best fit for, you know, KCA, not just to have access to the data, but also to provide access to the data to the community. Concomitant to the data is also access to tissue, which is very important. I want to just put your attention here to a very recent announcement. We did a partnership with the Rare Cancer Research Foundation at Pattern.org. If you don't know, Pattern.org is an organization that allows for patients to participate in research, donate their excess fresh tumor tissue. They have already protocol in place with, you know, shipping kits where they can be delivered directly into the operating room, and with patient consent, take part of this and ship to the investigator that's really requested the tissue. So the patients can actually say what they want their tumor tissue to be done with. So we are very excited by these options. We're going to include it in, as an option for the PIs to include it in their KCI grant. So they can use that for, if they need access to fresh tissue. Last but not least, I would like to, have here on my slide another very important piece of our collaborations. Everybody in this room is probably, representative of one of these companies. We need industry. We need, companies that can actually cross and take those treatments to the patients. Their commercial capabilities, their scalability, research is great, but without industry, we couldn't really bring those to the patients. So thank you all, and I'm really excited by the breadth of different players, and each one of you brings something important, because, of course, we're all driven by the fact that we want to bring new treatment, new therapies to patients, and better quality of life for all kidney cancer patients. So with that, thank you so much. I hope you learned something, and I'm here for questions. If any. Otherwise, gladly, grant some extra time to the panel. Thank you so much again.
Thank you so much. So we're going to move right into probably one of the greatest success stories we've had in recent years, the belzutifan story. Starting off with Dr. Bruce Posner, PhD at UT Southwestern with extensive expertise in drug discovery, high throughput assays. And he's going to talk about the preclinical story of belzutifan from basic research insight to clinical candidate.
Well, let me just start by thanking the organizers for the opportunity to be a part of this the symposium and be able to represent the UT Southwestern team today. As many of you know, basic research insights can have impact on therapeutic approaches, and the same is true for belzutifan. Our approach really began with two investigators at UT Southwestern, Rick Bruick and Kevin Gardner. And both of them shared an interest in PAS-B containing proteins, in particular the HIF family of transcription factors. These transcription factors, as you probably know, are upregulated during low oxygen conditions, and when they are activated, they turn on hundreds of genes that enable the cells and tissues to survive under these conditions. But it's absolutely critical that these transcription factors be shut off when normoxic or regular oxygen conditions are restored. Otherwise, if that's the case, then in fact we get unregulated expression of those transcription factors, and we can have a consequence in which unregulated growth can occur. Now, some PAS-B domains are regulated by small molecule cofactors, and this provided some impetus for the study of the HIF-2 alpha protein. It's a known driver of VHL-associated disease, and genetic ablation of HIF-2 alpha impedes tumorigenesis in renal cell carcinoma. The question really was for these investigators, is a transcription factor druggable? Many people in the pharmaceutical industry had said that, in fact, this was an undruggable class of proteins. However, a key insight came from structural studies of the HIF-2 alpha protein. In particular, these two investigators noted that there was a 290 cubic angstrom cavity inside of the PAS-B domain of the HIF-2 alpha protein, and that, in fact, this cavity could be exploited to alter the affinity of the HIF-2 alpha protein for the HIF beta protein. So they envisioned an antagonist that could reduce HIF target gene expression and therefore disrupt tumorigenesis. So in designing the assay, it was a very simple approach. We looked simply at the PAS-B domains. Those are the domains that are highly regulated in many of the alpha subunits or the alpha proteins for HIF family. And in this case, we linked the green protein, which is the HIF2 alpha pass B domain, to a donor bead, and the HIF1 alpha or ARNT pass B domain to an acceptor bead. And this is fairly simple conceptually. When those two proteins are close to one another and we shine light on the donor bead at 680 nanometers, it generates singlet oxygen, which travels to the acceptor bead, and we see light emanate from there in the form of a fluorescence cascade, which is readily measured. So the concept of the assay was fairly straightforward. If we disrupt this by binding a small molecule at the pocket that Bruick and Gardner had identified, we could hopefully alter the conformation of the HIF2 alpha pass B domain and interrupt its association with the HIF beta or ARNT subunit. So any drug discovery effort, even in academia, is a multi-investigator approach. My group worked with Rick and Kevin on a high-throughput assay, which we optimized, and I just showed you in the previous slide. And then we ran a chemical library screen against our institutional library, which at that time was about 200,000 compounds. We identified about 3,500 hits, that is, compounds that were statistically significant with respect to disrupting that complex. And from there, we conducted multi-dose confirmation studies with the primary assay and counter screens, which basically whittled this list down to a handful of compounds. And that's when we engaged Drs. Tambar and McMillan, as well as Dr. Williams. Tambar and McMillan are medicinal chemists by training, and they helped us optimize that initial hit structure into a lead molecule, and Dr. Williams helped us optimize for ADME properties. And that ultimately led to a lead molecule with a KD, or ability to disrupt half of that complex at around 70 nanomolar. Now, importantly, Dr. Gardner was able to show that this compound does in fact bind to the pocket that was identified previously, and in doing so shifts the conformation of the HIF2 alpha pass B domain such that it is unable to bind to the HIF1 HIF alpha protein. Although that is very interesting data to have, we wanted to show that this is functionally relevant in kidney cancer cells, and we used this compound to dose those cells and then basically lyse those cells open and assess whether a complex had indeed formed. We can use antibodies against the ARNT protein to pull down that protein as well as any other proteins associated with it. Now, hopefully you can appreciate from this Western blot that as we increase the amount of compound, in this case, the amount of HIF2 alpha protein present decreases as a function of dose. The HIF1 alpha protein, a sister protein to HIF2 alpha, does not have this pocket and does not change in its concentration as we expose it to compound. So this is a good proof of concept. We wanted to take this further and show that in fact we could interrupt the association of these two proteins on an actual response element for a target gene of this family. And that was the EPO erythropoietin gene response element, and we were able to use a chromatin immunoprecipitation assay to show that specifically under low oxygen conditions, there was no change in exposing these cells to the compound in the association of HIF1 alpha with that particular response element. However, you can see a dramatic effect when we do that with the HIF2 alpha protein. Unfortunately, I don't have a pointer, so I can't really get this to you, but hopefully you're able to pull that out just without that. So that led to a license with a company called Peloton Therapeutics. Peloton was led at that time by John Josie, and they took our lead, as you can see in the upper left, and they applied medicinal chemistry, structure-based drug design, biology, ADME, and all of those necessary components for drug development to identify a clinical candidate, PT2385. As you can see in figure 3 here, that in a dose escalation study with this compound, we had reasonably good results. 52% of the patients showed stable disease. There was 1 complete response, and 12% of the patients had partial response. In a companion study that was done by Kevin Courtney and Jim Bergolis at UT Southwestern, they were able to take biopsies from these patients both prior to treatment and on treatment at 7 weeks, and show that in fact that the compound was able to disrupt the complex, and that's what's shown in this figure 2. Hopefully you can appreciate the red dots that are showing on these cells. Those represent the formation of that complex between HIF2 alpha and HIF beta. And on treatment, you can see that 2 of the 3 patients showed a statistically different level of that complex, the third patient showed a trend in that direction, but not statistically significant. Unfortunately, 2385 had some problems with exposure. It was quite variable across patients and was determined by Peloton that, in fact, that was due to glucuronidation of that compound. The glucuronidated addict was not active against the HIF-2 alpha, HIF-beta complex. And so they went back to the drawing board and designed PT2977, which ultimately became belzutifan. And I hope you can appreciate that it was a simple adjustment, moving the fluorine, you can see in the ring there, from a geminal position at the same carbon to a carbon next door. And that obviated completely the glucuronidation and allowed for greater exposure in patients, more consistent exposure. And ultimately, when this compound entered phase 2 clinical trials, the overall response rate among RCC patients was 49%, with a 56% duration, having a duration of response greater than 12 months. Throughout the process, UT Southwestern stayed engaged in this program, both with Peloton and with Merck. And fortunately, in August of 2021, this compound was approved as belzutifan and has since continued to go through clinical trials. I believe that you'll hear more today about the LightSpark trials, which are extending the impact of this compound beyond the current indication. So the take-home here is that bench to bedside does in fact happen, and in this case it's a first-in-class medicine. An extremely rare thing among drug discovery folks. If you're in industry, I was served at Pfizer for several years. First-in-class medicines are quite special, and we feel very fortunate to have been a part of this process. I hope as you think about drug discovery targets, you'll challenge the concepts that people put forward about what's druggable and what's not, and clearly in this particular case, that paid off. It would take forever to acknowledge all the people involved. Here, it would take more time than I'm allowed. I'm already over, but simply to acknowledge that this is a huge effort, and we're so happy that everything has proceeded so successfully. And these are the full reference citations in case you're interested in studying more details of this particular story. With that, I'll thank you for your attention.
Thank you, Bruce. Well, that was the first step. Then we handed it over to the people who had to design it into a clinical trial. Dr. Eric Jonash from MD Anderson is here to talk to us about the IND package and early clinical development.
I want to thank the organizers for the opportunity to present this, and it's a real privilege. Obviously, this is a work of hundreds of individuals over the years, and we've got a great introduction in terms of the background chemistry here. Just from a standpoint of where this came from a VHL disease perspective, you know, already over 100 years ago, observations made by von Hippel and Lindau of this disease that we now call von Hippel-Lindau disease. In 1993, two things happened, I think, that were of equal importance. First was that the gene was discovered. The second was that the VHL Alliance, which was then called the VHL Family Alliance, was started, and it really was that ongoing partnership between patient groups and the scientists and the clinicians in the ensuing decades that I think gets us to where we are today. So you don't need the introduction here that, of course, it would be great if we could block HIF in those VHL mutated cells, and that would then affect the microenvironment in a positive way. And as you've heard, it was hard to create drugs that could do that. This small molecule that was developed, beautifully summarized, is modified in a very simple manner, and this paper in the Journal of Medicinal Chemistry in 2019 is actually really cool to read in terms of seeing how this evolved. This exposure of PT2385 as a function of the glucuronidation really ended up being a major issue, and because of these very simple modifications, a more effective drug was developed. So in terms of the development strategy, and we have Naseem Joswala, who's on the podium here with me. She was at Peloton, and this is a drug that was moved into this company called Peloton, and then went into early development, and then the compound and the company were purchased by Merck. And so the development really ended up being to early development with a small company, and then the later aspects of this development were then performed by Merck. And I'll sort of point out where this sort of handover occurred. So the first study was this LightSpark 001 study, which is what it's now called, which was a all-comers study, but with an emphasis on renal cell carcinoma. And the idea here that it would be all-comers just to make sure that there wasn't, for serendipity purposes, that they wouldn't miss signals from an efficacy perspective in non-renal cell carcinoma patients. And then there ended up being the extension arm of that study where we ended up enrolling the 55 patients with clear cell renal cell carcinoma. Moving on from there was a registrational study in VHL disease once the phase 2 dose was discovered. And then there was an analysis really of the various toxicities that arose as the preclinical data as well as the early clinical data came through, and a strategy that was articulated fairly early on with the FDA on how to actually manage these toxicities. The toxicities that were seen were testicular atrophy in rats and not in other animals, which was interesting, but to mitigate this, semen analysis was performed in male VHL patients early on, and fortunately there was no evidence that this was replicated in humans. The anemia and on-target effect was seen early on already in the early clinical development of this, and an algorithm for management was proposed and sort of evolved in the studies as they moved forward. And the hypoxemia, which is already seen with PT2385, was something that also was addressed head-on, and an algorithm for dose adjustment and dose reduction was incorporated into the belzutifan studies. This first study, the MK6482, which it was called when Merck took over, was like I said, all comers, but then the dose expansion was in renal cell carcinoma. We saw that the recommended phase 2 dose was 120 milligrams, although you could go higher, and there really did not look like there was a dose-limiting toxicity here. And with this study, primary endpoint was maximally tolerated dose or biologically reasonable dose, and then secondary endpoints of safety, PK/PD, and anti-tumor activity. Here are the inclusion criteria for this study. And the baseline characteristics of these patients, as we can see here, the dose escalation cohort, typical age of 63 years, the RCC cohort of 62. For the RCC patients, these individuals had a median of 3 prior therapies, and they had all the different types of therapies that one would expect at that point in the clinical sort of spectrum of therapies for renal cell carcinoma patients. The MTD, as I mentioned, was not reached. No DLTs were observed. And just based on safety and PK/PD, 120 milligrams was chosen. The initial data, and that formed the basis of the publication in Nature Medicine, showed that there were treatment-related AEs leading to treatment discontinuation in a relatively small percentage of individuals. And this is borne out, I think, in the future. We see that this drug is very well tolerated. There were no deaths due to treatment-related AEs. In terms of the side effects that were seen, it was anemia, again, not surprising. And we can see here fatigue, dyspnea, hypoxemia, and a few others. In terms of objective response rates in this this phase 2 study with 55 patients, we saw that the objective response rate was 25%. The PD, interestingly, was only 15%. And these responses seem to occur both in favorable as well as in intermediate and poor risk patients. Here are the waterfall plots, and we can see here again, this is not curing cancer, but it is definitely helping a significant percentage of the individuals who were on this trial. The other thing is that ongoing treatment was occurring in a subset of individuals, and so clearly this drug is helpful in a subset of individuals more than others, and even to this day we don't know exactly who these patients are, and that's a critical unmet need for the field. The progression-free survival was 14.5 months, which was pretty impressive for this heavily pretreated group of individuals. The plasma concentrations varied, but were definitely sort of as expected. I'll skip this one here. So with these data, the recommended phase 2 dose of 120 milligrams was then used to move into the registrational study for VHL disease. And here, genetic germline proven diagnosis of VHL disease was necessary. Patients had to have at least one measurable RCC lesion measuring at least 1 centimeter in diameter, no prior anti-cancer therapy. So this is really important because just from a registrational strategy, this was a registrational study. 61 patients ultimately were enrolled. It was powered actually to get its data with 50 patients. It was over-accrued to, but the other lesions were not actually in the primary endpoint. However, the secondary endpoints of looking at this in non-RCC lesions was heavily emphasized, and data collection occurred in a way that those data could be used potential for future registration in, for example, pheochromocytomas, pancreatic neuroendocrine tumors, the hemangioblastomas, both retinal and central nervous system. And that was a really great decision because that then allowed the label that we see later on. We saw that this study had an objective response rate of 49% initially, and this has gone up in the renal cell carcinomas of these individuals with von Hippel-Lindau disease. You see the waterfall plot on the right. PD as best response was seen in no patients. The longitudinal change in lesions over time was something that was of great interest to the FDA, because the argument here is, well, okay, these individuals have slowly growing tumors. Show us that you're actually seeing a salutary effect with this agent. And with these plots, with these spider plots here, we can see there's this growth over time leading up to initiation of therapy, and then there's clearly an inflection point and a reduction afterwards. And to everyone's great, happiness and to some degree surprise, we saw that there was objective responses in the pancreatic neuroendocrine tumors, in the CNS hemangioblastomas, and also in cystic lesions. The responses in the retinal lesions was also present, and there was stabilization and to some degree some shrinkage. And from a procedural perspective, this was another figure that we worked really hard on to show the effect on individuals that were living with VHL. We can see this vertical line. On one side, the number of procedures on a per patient basis leading up to the initiation of treatment, and then the number of interventions after initiation of treatment. A striking difference, obviously subject to bias, but I think reflective of what was happening with these patients. Adverse events were as expected. We saw anemia. In the VHL patients, not that much hypoxemia, perhaps because they're younger, because they don't have as much effect already over with just the time and disease on their lungs. And we saw that the anemia was something that stabilized over time. There was tachyphylaxis that developed. A number of these individuals received ESAs. The paper was published, and the approval occurred in August of 2021. And to our delight, it was for renal cell carcinomas as well as for central nervous system hemangioblastomas and pancreatic neuroendocrine tumors. So I think the lesson learned here is that when you're doing these early phase studies, gather more data rather than less data, because it might help you. So there's a robust development strategy here. The initial studies really were the 001 study, the 004 study, and the 003, which is not on here, which was the cabo plus belzutifan. Those were initiated by Peloton. Then when Merck took over, then the 005 study, which we heard about at ESMO this year, was then further modified by Merck and opened up. And then there was basically then phase 2 of the next line studies, 11, 12, and 13, 13 being dose optimization, 12 being a basket, combining with TKIs and with IO therapies, and 11 being a registrational study of belzutifan plus lenvatinib versus cabozantinib. And then there's the newer phase of studies here, which would be 15, which is in FIOS and PARAS, 22, which is the adjuvant study, and 24, which is together with palbociclib. So in summary, obviously this seminal observations by bench researchers that led to the understanding of how VHL regulates HIF, then led to UT Southwestern's development of a compound. For some reason, this is clicking forward. And then we get the approval after much work in 2021. And there's obviously a lot of work to do. We don't know exactly how to combine this drug with other agents. We don't exactly understand what does HIF-2 alpha blockade do on the various immune subpopulations within the tumor microenvironment, and there's efforts underway to learn this. Acknowledgement of many, many people, and this is obviously a completely incomplete list of people who made this possible. So thank you very much, and once again, what an amazing journey for the scientific community and for patients.
Thank you, Eric. Next we have Dr. Jalifala. I hope I'm pronouncing that correctly. She is a medical oncologist who works for the Center for Drug Evaluation and Research at the FDA, and she's going to talk to us about the FDA evaluation of the data and final approval for belzutifan.
Hi everyone, my name is Rala Falla, medical oncologist at FDA, and today I'm going to present the FDA's evaluation of data and approval for belzutifan. Oh, sorry. Oh, okay. So first I'm going to give some background about VHL disease tumors. It's a rare disease and caused by a germline mutation in the VHL gene, and affected patients are at increased risk of developing a variety of benign and malignant tumors, which are listed here. RCC develops in up to 70% of patients with VHL disease, and the risk of metastasis is correlated with the tumor size. So to prevent metastasis, surgery is recommended for RCC tumors of 3 centimeters and larger. About 10% of pancreatic lesions in patients with VHL disease are neuroendocrine tumors or PNETs. These are also malignant tumors, and surgery is indicated when tumor size is 2 centimeters or larger in the head of the pancreas, or 3 centimeters or larger in body or tail of the pancreas. CNS hemangioblastomas in the blue box are benign tumors which can cause serious morbidity or mortality in patients due to the mass effect. And surgery is indicated when these lesions become large and symptomatic. Retinal hemangioblastomas are benign tumors in the eye, which can affect patients' vision. And local therapies such as laser photocoagulation are the preferred approach for management of these lesions. And there are also other tumors such as pheochromocytomas and paragangliomas, pancreatic cysts and cystadenomas, endolymphatic sac tumors of the ear, and cysts of epididymis and broad ligament. So the application that was submitted to FDA for belzutifan was based on LightSpark 004, which is a single-arm trial that enrolled patients with localized VHL disease-associated RCC. Patients received belzutifan at a dose of 120 milligrams once daily, and the primary endpoint of the study was objective response rate in the RCC by Independent Radiologic Review Committee, or IRC, per RECIST criteria. After the study enrollment was completed, the protocol was amended to add retrospective IRC evaluation of existing images for CNS and retinal hemangioblastomas and pancreatic lesions, including PNET. With a median duration of follow-up of 22 months, the confirmed objective response rate for RCC was 49%. Median duration of response had not been reached, and more than half of the patients with an objective response had an ongoing response at 12 months. An analysis of the linear growth rate of the RCC on study 004 showed an increase in tumor size before and a decrease in tumor size after initiation of belzutifan. The sponsor also conducted a retrospective evaluation of the VHL database at NIH, and the median linear growth of RCC tumors was 3.7 millimeter per year in that study, which is similar to the pretreatment numbers in study 004. These two analyses provided supportive evidence that the slowing of RCC tumor growth on study 004 was due to the treatment effect of belzutifan and not a spontaneous regression of the tumor itself. The retrospective review of CNS images by IRC identified 50 patients with CNS hemangioblastoma at baseline. However, we noticed that these measurements included both solid and cystic lesions, as well as the edema around the tumors. So we asked the sponsor to review these CNS images, this time excluding cysts and edema from their measurements. And 24 patients were identified with at least one measurable solid CNS lesion at baseline. Of those, 63% had a confirmed objective response, including one patient with a complete response. And more than 70% of those with a response had an ongoing response at 12 months of follow-up. The same images that were initially acquired for RCC were then retrospectively used for assessment of pancreatic lesions. So the images were not optimized for detection of PNET on a number of parameters, including contrast timing and slice thickness. And this led to a high level of discordance in diagnosis between the investigator and IRC, as well as between the IRC readers, which needed further adjudication on a high proportion of patients. According to the application submission, 22 patients had PNET at baseline per IRC assessment. However, after adjudication, only 12 patients had a measurable PNET at baseline per at least 2 blinded radiologists. Objective response rate in those 12 patients was 83%, including 2 complete responses, and half of those with a response had an ongoing response at 12 months of follow-up. For assessment of the data for retinal hemangioblastoma, we consulted FDA's Division of Ophthalmology, and according to their review, of 28 eyes with available retinal images, efficacy could not be assessed in 23 eyes for various reasons such as lack of disease at baseline or use of laser treatment. 5 eyes with retinal hemangioblastoma at baseline seemed to have improvement in size of lesion per IRC assessment without potential confounding circumstances. In study 004, reduction in size of benign pancreatic lesions was seen, but the interpretation of results was limited because of the high level of discordance between investigator and IRC assessment and potential for spontaneous regression and resolution of these lesions. 3 patients with pheochromocytoma and 1 patient with ELST had stable disease per investigator assessment, and of 10 patients with epididymal cystadenoma, 2 had improvement in size of lesions and 8 had stable disease. In study 004, the median duration of exposure to belzutifan was about 16 months, ranging from 2 to 24 months. There was no fatal adverse reactions. Serious adverse reactions occurred in 15% of the patients, and discontinuation rate due to adverse reactions was 3.3%. Anemia occurred in 90% of the patients on study, but it was mostly low grade. And 1 patient had hypoxia, which was grade 3. So the main factors we took into consideration for decision-making was that this study evaluated patients with a rare disease in an early non-metastatic setting, and conducting randomized trials with time-to-event endpoints in each of these tumors is extremely challenging. In addition, morbidities such as risk of metastasis, neurologic deficit, and need for surgery in these patients are correlated with the size of the tumors. And we knew about the biology of the HIF2 alpha pathway and the shared pathophysiology across these tumors. So with these considerations, the FDA concluded that there was a favorable benefit-risk profile for belzutifan in VHL disease-associated RCC, CNS hemangioblastoma, and PNET. The magnitude of response and likelihood for symptomatic improvement, long duration of response, biologic rationale, and rare tumor type allowed for granting a traditional approval. For other VHL tumors, the data were not adequate for approval, and those tumors will be further assessed in more post-marketing studies to describe benefit. Another point to mention here is that the degree of embryofetal toxicities with belzutifan was similar to many other approved oncology drugs. However, patients with VHL disease are young. In study 004, the median age was 41, ranging from 19 to 66. And these patients will be exposed to belzutifan for a long duration. In addition, belzutifan may render some hormonal contraceptives ineffective. And based on these concerns, we added a boxed warning to the label for embryofetal toxicity. And the last topic, management of anemia. In study 004, anemia was managed with erythropoietin-stimulating agents, or ESA, and blood transfusion. 12 patients received ESA and 5 patients received blood transfusion. There are some warnings for ESA use due to potential for increase in risk of death, tumor progression, thrombosis, myocardial infarction, and stroke. And in LightSpark 004, the safety data for ESA was from a small number of patients with limited duration of follow-up. So in the dose modification section of the label, we recommended blood transfusion instead of ESA, while the safety of ESA use in this setting has not been established. And also, we recommended a higher than routine hemoglobin threshold of 9 for dose interruption and dose reduction to reduce the potential need for blood transfusion. In addition, a post-marketing requirement was issued for longer follow-up of safety on study 004, which will provide additional data on safety of ESA use in this setting. And that concludes my presentation. Thank you for your attention.
Thank you so much, Shawat. And finally, we are going to hear from Dr. Naseem Jawala, who has been involved in this since the very beginning with Peloton, just to give us her reflections. Excuse me? You're welcome to be wherever you're comfortable. Just to give us her reflections on the drug development pathway.
Thank you, Jody, and thank you all for inviting me to this symposium. So just a little bit of background about myself. I'm a medical oncologist. Currently, I work at a company called Olima Oncology, which focuses on breast cancer. But prior to that, I was at Peloton Therapeutics, and I actually started at Peloton in 2015. I was the first physician hired to the company and was really the only physician for a number of years there. And I think why that's important is the development of belzutifan really reflects a collaboration of industry, academia, clinical medicine, and patient advocacy, because at a small company, we just didn't have a lot of people, and I really had to lean on the clinical investigators early on to help me elucidate the safety and efficacy of a first-in-class agent. So just some milestones I can point out. The very first patient that was treated with 2385 was actually in Nashville in February 2015. Here we are. And the ESMO presentation given earlier last month in Madrid by Laurence, actually 5 years to the date prior, I had sat in a room with her with a number of other members of the steering committee to design that study. So it's really great to see how this has all come about. You know, there were a lot of challenges early on, as Bruce and Eric talked about, with the pharmacokinetics of the drug, elucidating the hypoxia and that adverse event and how to treat it was not only challenging as us as company members in dealing with the clinicians and also in discussions with the FDA. So, you know, it's been a long time coming, but, you know, it's really been from bench to bedside and approval to see it all come together. It's almost brings tears to my eyes. So I'm happy to take questions about, you know, early development, but really thanks to all of you for bringing this drug to market and to patients, and hopefully soon to RCC patients as well as the VHL community.
From the floor? All right, thank you very much. I think Gretchen's coming up. Oh, I'm sorry.
Time for questions. Okay, Yusuf Zakaria from University of Iowa. Great session, and shows how much it takes a village to make a drug. So I have a story of a patient, VHL patient, referred to my clinic, no kidney cancer, referred by ophthalmologist, actually, who suggested that only had retinal angioma and no other indication for belzutifan, and referred specifically for belzutifan. So there's no indication. We went through insurance denial. We went through colleagues in pharma. Initially, a lot of roadblocks. So the comment I received from ophthalmology: if I do any focal therapy, patient will be blind, and this is a young patient, VHL. So after long back and forth, basically we were able to get the belzutifan, and patient is in great shape now. Great response, able to see. So my question to Dr. Fallah, and a plea to our pharmacy industry colleagues. So from your presentation, you mentioned you did not have enough data for retinal, right, to evaluate. Isn't it fair to assume that those patients might benefit from it and offer it FDA approval? And my plea to my industry colleagues, can we make it easier on us to have access to these drugs to our patients when we are able to extrapolate and we think it will help our patients? And we have a lot of examples of this. Thank you.
I'm sure that many people have the question about retinal hemangioblastoma, and we had a lot of discussions internal discussions at FDA. So your example was an exception. Your patient was not eligible for laser photocoagulation, and it could make the patient blind. So that's a different story. But for most of the patients who are eligible for laser photocoagulation, we need to evaluate the risk and benefit of each of these treatments. Belzutifan is a systemic treatment. Still, it has low toxicity profile, but still there are toxicities, and patients are at risk of, they may need frequent blood transfusions. So there is a systemic treatment with adverse events, compared to a local treatment, patient can go to the doctor's office, get local therapy, and then without having any systemic toxicities. So for most of the patients who are eligible to receive that local treatment, we need to evaluate and compare the risk and benefits. And we could not answer that question with the results from study 004.
Who's first? Go ahead.
Question to the preclinical package. I see a lot of emerging targets now in biology. The idea is how can we find the next to fund that is targeting specific and having a different drug in our armamentarium that is in that direction and not the next TKI that is a general drug that hits many targets. What was the key element, I was wondering, of preclinical data that convinced clinicians to move this into patients? Can you specify, is the robustness of the in vitro results a specific animal model? And what is the real package? Is like the reliability of animal models that that are relevant more than anything else that convince clinicians to move this into patients?
I mean, I'm going to try to answer that. I think it was, what we have here is we have a very clear mechanism of action. We have a very, very clear pathway. We have a very clear mechanism of action. There were very clear preclinical data showing that there was target engagement and you were getting the desired effect. And so all of these things together, I think it provided, the clinical hypothesis that this would actually translate into therapeutic benefit. And also, I think in terms of the preclinical toxicity profile, this ended up not being, with the exception of the potential testicular toxicity in that, all of these were fairly expected. And I think that in aggregate resulted in the decision to move forward to clinical testing. That would be my perspective on your question.
And just to add to Eric's analysis, that very early on when we treated the first few patients, immediately we would look at the erythropoietin levels because that would demonstrate target engagement. If the EPO was dropping, we knew the drug was working, and that was really based on the preclinical data where we'd seen that.
Last question, Laurence.
It's actually more a comment. First, I want to congratulate the organizing committee for this session. I think it's amazing for all of us to have new drugs coming up, not only for VHL, but also for sporadic RCC. I wanted to highlight where KCA and advocacy group actually played a role in this journey. One of them is that it is within this meeting, actually the European one, that we were among the first to have the access of the clinical data being presented to our community, and I think that also spread the word within the RCC field to see the activity we had beyond VHL. And the second thing is about patient advocates. In the US, you do have access to belzutifan. I'm not sure you're all familiar with the fact that EMA didn't follow the same decision as FDA, and we don't have access for our patient with VHL. This is changing, but it is changing thanks to the patient advocates who clearly made the push. And I think it's important to highlight that sometimes authorities may not be as convinced with phase 2 non-randomized data. And this is where we need patient advocates. So thanks again.
Thank you.